Welcome back to your Pediatric Dentist channel Tiny Teeth Talks. If you are new to this channel, subscribe the channel, and for details of this channel visit the bio section, or you can also check out my first video. We already discussed how the tooth erupts with the theories of tooth eruption. Today we will discuss the shading of deciduous teeth. Human dentition consists of two generations. The first is known as the deciduous dentition and the second is permanent dentition. The necessity of two dentations exists because infant jaws are small and the size and number of the teeth they can support is limited. Since teeth once formed cannot increase in size, a second dentation consisting of larger and more teeth is required for the larger jaws of the adult. Therefore, the resorption caused by the pressure from the erupting permanent tooth and the disintegration of the roots and supporting structure weakens the tooth and cause exfoliation. The physiologic process resulting in the elimination of the deciduous dentition is called shading or exfoliation. Pattern of shading involves two patterns that works together. First, resorption of roots, progressive breakdown of the roots and supporting tissues of the deciduous teeth, and pressure from permanent teeth. The erupting permanent teeth exert pressure dictating the pattern of resorption. Now let's see how eruption works in two different regions, which are anterior region and posterior region. Let's start with anterior region. Permanent anterior tooth germ is located lingual to the apical third of the root of the corresponding primary tooth. Here is permanent central incisor located behind the primary central incisor. The resorption begins in an occlusal labial direction which corresponds to the movement of the permanent tooth germ during its eruption, so that permanent tooth take position. As the crown of the permanent tooth moves apically closer to the root, resorption progress horizontally to allow the permanent tooth to erupt into the position of the primary tooth. So the permanent anterior tooth germ located behind the primary tooth, resorption in occlusolabial directions because the permanent tooth erupts in that direction, and as the crown come closer there is a horizontal root resorption so that permanent tooth can take their position. When we talk about posterior teeth, it includes only premolars because molars don't have predecessors. The crown of the developing premolars are initially situated between the roots of the primary molars. Resorption start with the interradicular bone, which is the bone between the roots of the primary molar, and continues with the adjacent surface of the root. Then the alveolar bone grows and adapts to support the lengthening roots of the permanent tooth, ensuring the proper alignment and eruption process. As the resorption progresses, the primary molars eventually lose their support and exfoliate, allowing the premolars to emerge in their place. So in posterior teeth, resorption start with the interradicular bone because of the crown of the premolar situated there, with the growing alveolar bone support the root lengthening of the permanent tooth, and eventually primary tooth lose support and exfoliate. Let's understand this procedure in detail with the mechanism. There are three reasons for resorption and shading: the loss of roots, loss of bone, and increased force. Let's see how. Kronfeld was one of the first researchers to suggest role of stellate reticulum and dental follicle in shading mechanism. Let's see how. So as the permanent tooth grows within the alveolar bone, it exerts the mechanical pressure on the surrounding tissue and roots of the deciduous tooth, which include bone and root, which stimulate the cellular and molecular responses, which include the role of stellate reticulum and role of dental follicle. The pressure is a critical trigger for the initiation of the shading processes. The stellate reticulum is a star-shaped cellular network within the enamel organ of the developing tooth. It provides structural support and acts as a reservoir of signaling molecules. So here the stellate reticulum in the enamel organ, which is the star-shaped cellular network. They release the cytokines and growth factors. So cytokines are signaling protein, and different cytokines stimulate and differentiate different cells. These cytokines and growth factors stimulate and differentiate resorptive cells. They stimulate the differentiation differentiation and activation of the resorptive cells, which are osteoclast and odontoclast. The dental follicle is a connective tissue sac that surrounds the developing tooth germ. It contains the progenitor cells that can differentiate into the osteoclast, cementoblast, and fibroblast. So progenitor cells are committed to differentiating into a specific type of cell or a limited range of cell types. For example, hematopoietic progenitor cells give rise to the various blood cells. Osteoclast. So osteoclasts are the multinucleated cells that derive from the monocyte macrophage lineage. So this monocyte macrophage lineage refers to a group of immune cells that originated from hematopoietic stem cells in the bone marrow, and these stem cells differentiated into the myeloid progenitor cells, which further develop into the monocytes, and these monocytes are a type of white blood cells that circulate into the bloodstream and can migrate into the tissue. Once they enter into the tissue, monocytes differentiated into the macrophages or dendritic cells depending on the signals they receive from their environment. Monocytes derived macrophages fuse and form the osteoclast that is responsible for the bone resorption. They possess a ruffled border that increase the surface area of the resorption. So the ruffled border is a specialized, highly folded membrane structure on the side of the osteoclast that faces the bone surface. This unique feature significantly increases the surface area of the cell, allowing for more efficient bone resorption. The ruffled border is surrounded by a sealing zone which isolate the resorption area from the surrounding environment. Acid phosphatase released by osteoclast disrupt the collagen network, releasing crystals that are digested by vacuoles, which are membrane-bound compartment in osteoclast. The ruffled border forms a tight seal with the bone surface, creating a microenvironment which is known as a resorption lacuna. Within this lacuna, the osteoclast release this acid phosphatase, an enzyme to dissolve the bone matrix, which disrupt the collagen network. Acid phosphatase disrupt the collagen network, which is the structural framework of the bone matrix. This disruption weakens the matrix and facilitates its breakdown. Now, this bone matrix is composed of both hydroxyapatite crystal and the collagen components. As the collagen network is degraded, the embedded hydroxyapatite crystals are released, and these crystals are essential for bone growth and rigidity, and the release marks and breakdown of the mineralized matrix. Now, these released crystals and the collagen fragments are engulfed by osteoclast through a process called endocytosis, and once inside the osteoclast, the materials are directed towards the vacuoles, which are the membrane bound components within the cells, and these vacuoles fuse with lysozymes, which are organelles containing digestive enzymes like cathepsin K and acid phosphatase. These enzymes break down the collagen network and other organic components into the smaller fragments that can be further processed or expelled from the cells. Now, odontoclasts. So odontoclasts are specialized cells that play a crucial role in resorption of dental hard tissue such as enamel, dentin, and cementum, which is found near the resorbing root. The cells are guided to specific sites where resorption is needed, ensuring that the shading process is localized and efficient. So with the help of odontoclasts, which are responsible for the resorption of the dental hard tissue, and osteoclasts, which are responsible for bone and surrounding tissue, the resorption and shading occur. Forces of mastication are also synergistically involved in the mechanism of shading. As the jaw grow and develop, the muscles of mastication become stronger and forces exerted during chewing increased significantly. Also in Deciduous teeth, the periodontal ligament is less robust, less strong compared to permanent teeth. As the masticatory forces increase due to the jaw growth, they exceed the ligament's capacity to withstand stress. But these excessive force cause microtrauma to periodontal ligament, and these trauma trigger cellular responses in surrounding tissues, including the activation of odontoclast and osteoclast, and these resorptive cells begin breaking down the hard tissue of the tooth root and the supporting alveolar bone. Also, Eulambia herocopakis chisengalis introduce a concept that the shading of primary teeth is regulated similarly to bone remodeling, involve the rank or rankle, which is the receptor activator of nuclear factor kappa kappa B ligand system. This system is pivotal in osteoclast formation and activity. So first, the RANK, which is the receptor activator of nuclear factor kappa B, is a receptor expressed on the surface of the osteoclast and odontoclast precursors. It is essential for their differentiation and activation. Now, these precursor cells are intermediate cells that have partially differentiated and are capable of developing into specific cell type. And rankles secreted by the cells in the dental follicle, cell reticulum and surrounding tissue bind to this rank on the precursor cells and triggered intracellular signaling pathways that leads to formation of mature multinucleated odontoclast. As the permanent tooth develops, it exerts pressure on the surrounding tissue, stimulating the release of rankle from the dental follicle and other structures. Now, this rankle bind to the rank on the odontoclast precursors, activating the NF-kB pathway, which is the nuclear factor kappa B pathway, and this pathway induces the expression of genes necessary for odontoclast differentiation and activity. These precursor cells fuse to form the multinucleated odontoclast, which are specialized for the resorbing dental hard tissue. The activity of the odontoclast is conformed to specific area where the resorption is needed, such as the roots of the primary teeth, ensuring efficient shading. So the rank and rankle system ensure that the resorption process is tightly regulated and occurs only at the appropriate sites. It coordinates the activity of odontoclast with the growth and eruption of the permanent teeth, facilitating the natural exfoliation of the primary teeth. These are the references. If you want these as PDF or any other PDF, just like, subscribe and comment your mail ID with the topic and I'll send you the PDF. New videos on Sunday or sometimes Thursday and Sunday. Also, we try to put MCQs according to topic on our shorts with the answer and explanation if needed, which will help you in Viva or NEET exam. Like, share and subscribe the channel. Thank you.